Hello, everyone. I am Dr. María Yáñez Mo from Universidad Autónoma de Madrid, and I'm going to show you today some of our latest studies for the optimization of chromatographic techniques for the isolation of extracellular vesicles. Okay, so as you all know, extracellular vesicles have a complex composition that includes proteins, lipids, and nucleic acids, and have been involved in intercellular communication both in physiological and pathological conditions. So there's a lot of interest both in research but also in therapeutic applications of these EVs and in their use as in liquid biopsy for search for non-invasive biomarkers of disease. You also are aware that there are different kinds of extracellular vesicles, mostly two sorts, the ones that emerge from the endosomal compartments that are usually named as exosomes, and those that bud directly from the plasma membrane that are usually coined as microvesicles. But they share several aspects in their biogenesis, molecular details. So they share many also molecular components such as tetraspanins, and some of even the escort complex are involved in the generation of both types of vesicles. Okay, so I will refer in my presentation to extracellular vesicles because we isolate them from biofluids, both conditioned media or plasma, and it's therefore difficult to know whether they are derived from the endosomal compartments or they are microvesicles. So I prefer to use the general term extracellular vesicles. And as you know, there's different types of extracellular vesicle isolation techniques. The most common and the gold standard is differential ultracentrifugation. That is the most employed, but it has the drawback that with your vesicles, there will be a lot of protein aggregates, and it can also damage extracellular vesicles because of the high speed of centrifugation. There are also different kits and reagents for the precipitation of extracellular vesicles. These are very simple methods and very quick, but they have a very poor purity in your sample because they just extract water from the sample and everything will precipitate together, vesicles and non-vesicular components. And there are other higher purity systems such as immunoaffinity that are also quick and simple, and they will provide a high purity sample of extracellular vesicles, but they always be biased for the membrane protein that you use to immunoisolate the sample. Okay. Besides that, we can have a density gradient ultracentrifugation, which improves the purity of ultracentrifugation, but has a drawback of reducing the yield, and also size exclusion chromatography, in which the sample components are separated because of size. So the resin has small pores that allow the entry of small molecules into the pores, so those small molecules will be retained. They will take them longer to elute through the column, while extracellular vesicles cannot go into the pores, so they will directly elute out of the column. Okay, so both methods have higher purity. The size exclusion chromatography is suitable for any laboratory. You don't need an ultracentrifuge or anything. But in both terms, we really have to take care about lipoproteins, which is one of the major contaminants we might have in samples, such as blood-derived samples. And lipoproteins are very, very abundant, so they can be 1,000 times more abundant than the extracellular vesicles. And there are different kinds, chylomicrons, LDL, HDL, etc. And some of them will overlap in density, and some of them will overlap in size with extracellular vesicles. So that's something that we will have to take into account when using this kind of techniques. So we decided in collaboration with agarose bead technologies to improve size exclusion chromatography. And here there are different aspects of the resin that we can play around with. So we can use different polymer density from 2% agarose to 6% agarose. And this will impact on the pore size of the resin. And then we can also play around with the size of the beads in the resin. So there are smaller beads and larger beads. The larger the beads are, the fastest the elution speed will be. And then we can also use crosslinking, chemical crosslinking in the resin. And that is important for physical and chemical stability, mostly when you use very long columns or you don't use gravity elution, but you use ACTA system that implements pressure on the sample. Okay, so our methodology was first starting with conditioned media from a melanoma cell line. We do some centrifugations to remove cell debris, and we concentrate the conditioned media, and we perform the size exclusion chromatography with columns of 10 milliliters. We collect 25 fractions, and each of these fractions will be analyzed by dot blot or absorbance of protein content or Western blot. Also, we will do a nanoparticle tracking analysis and a transmission electron microscopy. You can find all this information in the manuscript we recently published in Journal Cellular Biology. Okay, so starting with polymer density, we analyze 2%, 4%, 6%, and 2% resin from a different vendor. And as you can see here, the 2% resin provides quite a clean extracellular vesicle. So the extracellular vesicle signal is the one in green and blue. While if you go to 4%, you have a little bit more of lipoproteins in the extracellular vesicle fractions, and this is very poor in the 6BCl sample. Okay, that you see that this orange peak overlaps very much with the blue green peak. Okay, so that is also analyzed, and so you have the 6BCl, which has a higher contamination by lipoproteins. The yield is a little bit higher, but the purity index is much worse than with the other resins. Okay, and these results are clearly revealed when we analyze these samples, the samples by transmission electron microscopy, in which you see lipoproteins as these small white dots in the background. So the 2BCl, the 2% resin, is cleaner than the ones with a higher percentage of polymer. Okay, so 2% is the one that most people used. The exclusion limit here will be around 17 nanometers, so everything bigger than 17 nanometers will elute first. But then we lose some of the smaller EVs, and if we use the 4BCl resin, the exclusion limit will be 35 nanometers, but then we will have some problems with contamination of lipoproteins. So we decided to play around with the other characteristics of the resins to see if we could improve this, but focusing on 4% resins. Okay, so we first changed the cross-linking of the beads, so we have different resins with different cross-linking, and as you can see, the graphs don't change much when we mess around cross-linking. But if we change the size of the agarose beads, then the things really improve. With the small bead resins, we can really resolve EVs from lipoproteins and soluble protein. And if you use a high big resin beads, then the thing gets worse. So with this small bead size resin, we do have very clean samples with very little contamination of lipoproteins, and that's also reflected in the quantitative analysis. And you see here the contamination is very low. The yield is a little bit lower, but not significantly different, and the purity index is much better than all the other resins we used. Okay, and that's also clear when you analyze by Western blot the lipoprotein contaminants with this resin here. Okay, so then we decided to move to human plasma. Here, the abundance of lipoprotein is much higher than in conditioned media from cell culture lines, but we do the same protocol. And as you can see here again, we compare the small bead resin. The lipoproteins are better resolved from EVs in the fractions, while in 2 BCl or 4 BCl, the contamination of EV fractions with lipoprotein is much higher. And this is also clear in the electron microscopy. You do see some lipoproteins in the background, but remember that you have 1,000 times more lipoprotein particles than vesicles in plasma. And finally, we wanted to compare with the gold standard, that is ultracentrifugation. And as you can see here, we had a very poor yield with ultracentrifugation compared with size exclusion chromatography. But then if you quantitate the protein in the samples, the ultracentrifugation gives a very high protein content, but this is not related to the number of vesicles, but with really protein aggregates that can be clearly seen in the electron microscopy micrographs. So we think that this improved size exclusion chromatography could make a big change in the use of extracellular vesicles for liquid biopsy. And when we talk about liquid biopsy, in most cases we would like to analyze nucleic acids, such as DNA or cell-free RNA. But RNA is also can be also analyzed in circulating tumor cells of platelets educated by tumors, but of course also in EVs. But we have also to remember that RNA can also be transported by lipoproteins or protein complexes in plasma. So with our improved resin, we wanted to analyze if we could detect a translocation, the RNA of the translocation of ALK, which is quite common in lung cancer patients. Okay, so for that we did again size exclusion chromatography, and starting with cell lines derived from tumors that are known to carry the translocation. And then with this size exclusion chromatography, we could interrogate separately the extracellular vesicles fraction, or the lipoprotein enriched fraction, or the soluble protein fraction of our conditioned media. Okay. And when we did so, you do see here that the positive signal of the transcript was detected almost exclusively in the extracellular vesicle component, and we scarcely saw some dots in the soluble protein or in the lipoproteins. Okay, so now we wanted to improve this with plasma from patients. Soon to come. Okay, so and finally, I want to summarize briefly another manuscript we did in collaboration with a group of Marina Kretic in Milan, and in which we improved affinity chromatography for EV isolation. And for the most part, affinity chromatography uses antibodies or aptamers against the membrane molecules on the EVs, with the drawback that there's no universal EV marker, so you will always have a biased sample as a return. So we decided to use the bradykinin-derived peptide that the group of Marina had shown in this paper published in Extracellular Vesicles that can bind to very highly pure membranes because it has amphipathic properties that allows the peptide to interact with the phospholipids in the membrane and with the holes that are created when this membrane is highly cored. So our scheme was to use, again, agarose resins with different cations that could bind to a polyhistidine tag that we hook to the membrane-sensing peptide. And then you attach the peptide to the beads, and then you add the conditional media with extracellular vesicles, and that's what you can easily isolate very pure extracellular vesicles. And you can do cargo analysis directly on the beads, on the EVs bound to the beads, but you can also very easily elute these beads, these EVs from the beads with imidazole or EDTA to do functional analysis. Okay, so with that scheme, we first compared the different resins with different cations, and we see that cobalt was the best one. And then we analyzed the concentration of peptide we needed, and we confirmed that there was no binding to lipoproteins to these resins. And also we optimized the time of incubation of the peptide with the beads and then of the extracellular vesicles with the beads. We also checked whether if we had a longer spacer in the resin, if that affects, and indeed with the longer arm resin we have better results of EV binding. And we also analyzed the different peptide architectures. So the one that we have been using so far is this tandem peptide, but we here analyzed a branched peptide that has 4 repeats of the peptide in this branched configuration. And the tandem peptide worked much better than the branched one. And finally, as I said, you can easily elute very gently the vesicles, and then you can do NTA and analyze them by transmission electron microscopy or perform functional analysis. And with that, I resume with the acknowledgements slides. I want to thank Beatriz Benayas, who is the leading force of all the work that I presented today. And this is the rest of my group. We did this in collaboration with the biotech company Agarose Big Technologies, and the last part was done in collaboration with a group of Marina Credits in Milan. So thank you very much.